organisms in general maintain homeostasis in different conditions. And so we mainly focus kind of post-transcriptional regulation. So I'm going to talk about a story which kind of my background. I was a chemistry undergrad major and then did biology PhD and went kind of to a systems biology lab for my postdoc. And that's where I got into using next-gen sequencing, and we would get 20 million, 100 million sequence reads. And so I had to pick up some limited scripting knowledge to be able to analyze this data. And I've recruited now students from bioinformatics backgrounds, engineering backgrounds, especially undergrads, to help with some of this analysis. And I'm going to talk about some of the work that some of the undergrads that have helped in my lab have done. So let's see. The central dogma is that if we're looking at a yeast cell, we have transcription takes place inside the nucleus where the DNA is converted to RNA, and then the RNA is exported to the cytoplasm where we have ribosomes engaged with the RNA and translate the RNA in the cytoplasm. And so my lab, I mentioned, kind of studies mostly post-transcriptional processes. We study mRNA localization and translational control primarily. And so translation has been studied at the level of initiation primarily because people have thought of this as kind of the rate limiting step in translation. But my lab is starting to become more and more interested in the elongation step of translational control. And so one of the things we were interested in, and I mentioned in the beginning, is how cells are able to respond in changing environmental conditions. So when things aren't normal, when things are more dynamic. And so in yeast, they generally grow on glucose and they're happy. And so this is a polysome profile, which I'm going to talk more about this, where a polysome is when you have a single mRNA can be engaged with multiple ribosomes, and those will sediment differently if you're using biochemical techniques. And so if we have one ribosome or two ribosomes, 3, 4, 5, 6, 7 ribosomes, this is what we can see. If we glucose starve the cell, we see a collapse in the polysomes, and we also see a large reduction in the protein synthesis as measured by radioactive amino acid incorporation. And so we wanted to use a global measurement of what's happening in these conditions, because we also know that when cells transition to a new media or a new environment, that they need to change their proteome to better survive in those new conditions. But if they're shutting down all translation, then how are they able to survive in these new conditions? And so to answer that question, about 10 years ago, a new technique was developed called ribosome profiling. And the premise is that you have these RNAs with multiple ribosomes on them, and then you use a nuclease digestion to digest all the RNA that's not protected by a ribosome. So all of this RNA would get digested, and what you're left with is this about 30 base pair fragment of RNA. You can then purify that RNA and generate a next-gen sequencing library. And again, this will give you can get like 20 million sequence reads. And so whenever I first was starting to get into this in my postdoc, generally it only used Excel, and there's nothing you can do with 20 million sequence reads in Excel. And so we had to develop Python scripts to analyze this data. At the same time that you're getting where every ribosome kind of is in the cell located on an mRNA, we also would get these total mRNA abundance measures. And so from this, you can get the ribosome and mRNA number of every RNA in the cell. And so here I'm just showing you example data for what things look like, where this is one RNA called hexokinase 1. And in glucose, it's generally repressed, and there's very few ribosome reads. Upon glucose starvation, we see this large increase in the reads that are along the RNA, and we also see an increase of ribosomes of ribosome protected reads, but it's not to the same magnitude. And so we can divide the number of ribosome reads by the total mRNA reads. So ribosome reads divided by mRNA reads, and this equals ribosome occupancy. And so generally people in the field had mentioned that ribosome occupancy directly correlates to the translational efficiency. If you have more ribosomes, the thinking is that those ribosomes are moving and you are having higher translational efficiency. But I'm going to tell a story where we found that this ribosome occupancy doesn't necessarily directly correlate to the moving ribosomes within the cell. And so if we do that, we did that for hexokinase 1, we can look at a single gene, we can look at this genome wide. And so here's hexokinase 1, where it has a large increase in the mRNA levels, as seen here. So the mRNA levels increase 30 to 40 fold. The ribosome levels also increase, but the ribosome occupancy levels decrease dramatically for this hexokinase 1 gene. And we can see the ribosome density over here. So hexokinase 1, some of these other genes, they show large increase in mRNA levels, but a decrease in ribosome occupancy, while other stress response mRNAs have increase in both mRNA levels and ribosome occupancy. But what was kind of surprising was that we saw certain RNAs that we usually think of as growth mRNAs, and that they had like PAB1 and PGK1, and that they still had high ribosome occupancy even during glucose starvation. And this was very surprising to us because we thought that most RNAs should be getting shut down translationally because the cells are stressed, and so that they wouldn't produce these housekeeping proteins for these housekeeping RNAs. And so we weren't the first ones to kind of see this ribosome still being present in the RNAs. A paper in 2005 also saw that for PGK1, even though there was some loss of ribosomes from this polysome, so again, here's plus glucose, you see PGK1 mRNA is generally very abundant in these high polysomes. Upon glucose starvation, you see some of the RNA leaves the polysome, but you still have quite a bit of RNA still engaged with multiple ribosomes, which again, we usually think of that as being highly translated. So another thing that we noticed when doing kind of genome-wide averaging is we looked at what other characteristics are related to ribosome occupancy. And we found that genes that were longer seem to have more ribosomes or a higher ribosome occupancy in glucose starvation. And so we can start to look at where the ribosomes are along the mRNAs, just using simple scripts to look at the distribution of ribosomes. And so in log phase, whenever cells are growing well, we see that the number of ribosomes, if we categorize them by either high ribosome occupancy or low ribosome occupancy, that the amount of reads right at the AUG correlates very well. So the ones with the most ribosome occupancy in plus glucose conditions also have the most ribosomes at the AUG. The ones that have the lowest ribosomes in the downstream region have the lowest at the AUG. And so this ribosome buildup at the AUG in log phase conditions seems to correlate with translation initiation. And so the more initiation you have, the more ribosomes you have downstream. But again, things get a little strange when we start looking at glucose starvation and the distributions of ribosomes in those conditions. So in glucose starvation, we have these upregulated high ribosome occupancy and upregulated low ribosome occupancy, like the hexokinase 1. And so both of those have lots of ribosomes in their AUG, but the blue genes like hexokinase 1 have very few ribosomes in their downstream region, while the red genes have lots of ribosomes in their downstream region. And what was kind of surprising was these housekeeping mRNAs, where they have very low ribosomes at their AUG, but they seem to have a high abundance of ribosomes in their downstream coding region. And this was kind of surprising whenever we think of how quickly ribosomes move, where if we thought of a housekeeping mRNA, it's highly abundant during log phase when there's glucose, and it's highly loaded with ribosomes. If we start to starve the mRNA, starve the cells, then translation, the ribosomes should run off if we shut off initiation. And so by a minute or 2 minutes, we'll have some partial runoff, but by 15 minutes, since ribosomes move at approximately 5 amino acids per second, we would assume that an RNA that's 1,000 base pairs or 300 amino acids, that would only take a minute or two to completely run off. But what we're seeing is instead that it looks like there's some slight runoff, but then that the ribosomes almost seem to stop and that there's not much runoff after some time. And so we're interested in looking at that more closely. And so we wanted to come up with a way to potentially quantify this in a way that's not just kind of looking at the so in this case, we had to aggregate a number of genes to look at the distribution of ribosomes. And so we can see the same distribution somewhat at the single gene level, but this data is very noisy. If we're looking at PGK1 and PAB1, you can see that ribosome profiling at the single gene level is somewhat noisy as you're looking at the distribution of ribosomes. So we wanted to use a way to calculate the distribution of ribosomes for every single gene. And so another lab had kind of come up with this polarity score. And so the polarity score is a way to calculate kind of the distributions of ribosomes across a single mRNA, where we have ribosomes all the way to the left would be a -1, ribosomes all the way to the right would be a +1. And so then we can get this kind of polarity at position I for every position where DI is just the ribosome density at that position. So DI equals and then WI equals the normalized distance. All right, so we can calculate the polarity at position I, and that's going to be weighted by how many ribosomes there are. So if we have lots of ribosomes here and a few ribosomes here, this is going to be weighted more heavily. And then we just take the sum of all these polarity measurements at each position. And so then we get a polarity for each gene. And what we can see is that for most genes in plus glucose, we see them as slightly negative in polarity. They have more ribosomes towards their 5' end than in their 3' end. And so people have seen this previously where there seems to be a slight buildup of ribosomes, especially in the beginning. And so if you're looking at the distributions of ribosomes, it would look something like this, where you have a slight elevation towards the 5' end, which gives you a slightly negative polarity. Now, if we look under glucose starvation, we see that for most mRNAs, we see a strong shift to the right in their polarity. They're starting to get a lot more ribosomes towards the 3' end of their RNAs. And so we thought this was a nice way to quantify the distribution of ribosomes within the cells. And one thing that you can notice is that there are some genes that have even higher polarity. And so we think these are the red genes, the HSP genes, where they're being transcriptionally upregulated and they're having a high amount of ribosomes initiate translation during glucose starvation. So whenever we looked at this delta polarity, we see that longer genes have a higher percentage of they have a higher delta polarity. So they have more of their ribosomes towards their 3' end than the shorter genes. So we think what's happening is that the shorter genes are actually running off and that the longer genes allow this accumulation of ribosomes to keep accumulating. And so the thinking would be then that what's happening is that the ribosomes are not elongating properly. So we think right now that this negative polarity is indicative of poor translation elongation. All right. And so we wanted to test that biochemically. So we're using these computational techniques to try and measure this globally and at an individual gene level, but we wanted to test this at the individual gene level. And so what we can do is we can look at these individual genes and their protein production in plus glucose. And so in plus glucose, the ribosome reads correlates pretty well to the amount of S35 that's incorporated. PGK1 has lots of ribosomes and it has robust S35 incorporation. PAB1 has lower ribosome counts and lower S35 incorporation. While HSP30 isn't really expressed that much in plus glucose. But where things get weird is whenever we have now this kind of positive polarity, right? Where PAB1 and PGK1, because it's kind of off scale, you can't see it as well, but they both have this distribution of ribosomes that increases to the right, and PGK1 has a polarity of about 0.2, and I think PAB1 is also somewhere around 0.2, while HSP30 is like negative 0.4 because it has most of its ribosomes are on its 5' side. So the RNAs that have this positive polarity, we see very little S35 incorporation. So PAB1, even though it has similar ribosome reads as HSP30, we can see robust incorporation. We see pretty much no new S35 incorporation during glucose starvation. PGK1, which has 25-fold higher reads than HSP30, actually has slightly lower PGK1 incorporation. All right, a question? Raised hand?
Yeah, I was just wondering what S35 is.
All right, so S35 is radioactive sulfur. And so what you take is methionine. Thank you for asking a question. So you can take methionine where the sulfur in methionine has been labeled with radioactive sulfur. And so but this will incorporate just fine into proteins as similar, just like a regular methionine. So you add this methionine to the yeast in plus glucose and you see how much gets incorporated into these different proteins. There's some added level of complexities where you have to purify specifically these proteins, but then you can use autoradiography to say how much radioactivity is in each band. All right. And so then you separate them on a gel, and we can say this is PGK1 incorporation, this is PAB1, and this is PAB1. Whenever we grow the cells without glucose, we add the same S35 methionine, and we see no S35 methionine getting incorporated into the PAB1 protein, even though there's plenty of ribosomes there. So we think what's happening is that this,
And just to confirm, PAB1 and PGK1 are basically the long genes that you're talking about, right?
they're long pre-existing genes. So they're housekeeping genes that are highly expressed before glucose starvation. But there are even longer genes, and we're going to kind of talk about them a little bit later, that have even stronger effects of accumulating ribosomes towards their 3' end. But yes, they are relatively long, but the bigger thing is that they're pre-existing before glucose starvation. All right.
Right, and can you repeat what's the role of these genes exactly, and how could that be related to glucose starvation?
So yeah, so PGK1 is a glycolytic enzyme, and so it's one of the key enzymes in glycolysis. And so if you don't have any glucose present, you wouldn't think you would need lots of glycolysis taking place, because the cell has already made tons of PGK1. And so now you're shutting off their glucose. They don't really need to make more PGK1. PAB1 is a translation factor that binds the poly A tail. It's again, the major thing is that these are highly expressed when the cell is growing very fast. And so the thinking is that they wouldn't need something like this during stress. But these heat shock proteins seem to help during many different types of stressful conditions. And it's unclear exactly what they're doing, but the general thought is that they can help like protein folding, and when things become misfolded during stress, when there's low ATP levels, there's even more problems with misfolding. And so HSP30 and a bunch of these other HSPs that we saw upregulated in glucose starvation can help prevent the stress from being too bad for the cells. All right.
Right, thank you so much. That
Yep.
makes sense. Yeah, it's interesting.
All right, and so one of the things we were wondering about is whether differential translation elongation is dependent on the mRNA sequence. Is there something uh, specific about the sequence, or could it be when it's transcribed that makes a difference? So we took an HSP30 promoter, which is only expressed during minus glucose, and a PAB1 promoter, which is mainly expressed pre-stress, and then we drove a luciferase construct. And so what we could see is very similar to what we saw with our S35 incorporation, where PAB1 is expressed highly during plus glucose and poorly during minus glucose. HSP30 is expressed poorly during plus glucose and highly during minus glucose. We see that PAB1 is expressed highly during plus glucose, but very poorly during minus glucose, while HSP30 is expressed poorly, but it's expressed well during minus glucose. But these are expressing the exact same protein. So we don't think that there's something to do with the codons that's driving this difference in elongation that we're seeing during the stress. And I'm going to talk a little bit about a hypothesis we have on what could be driving this difference. So one question we get is, so we think these ribosomes that have the positive polarity, where they're accumulating towards the 3' end, that they are paused. But then what's the purpose of, why would the ribosomes be paused during glucose starvation? And so one of the things we wondered is what happens to these ribosomes when glucose is re-added. So this is what we're doing, is we're just taking all genes that are above 5,000 base pairs and calculating the distribution of ribosomes across the RNA in plus glucose. And again, here you can see a negative polarity, and then the ribosome distribution in minus glucose, and you can see a positive polarity where a lot more of the ribosomes are distributed towards the 3' end. And so if we had kind of an arrow for where we start to see this ramp up in minus glucose, if we add glucose back, we can see that it hasn't really moved. 1 minute after we add glucose back, it hasn't really moved much. But then 5 minutes after glucose is added back, we see that this kind of ramp has moved a few thousand base pairs. And then we can also see this surge of new ribosomes coming in. So we think that this means that these ribosomes are stalled and not translating during glucose starvation, but that they can be translated upon glucose readdition. So they're kind of waiting there, since they're on these genes that are needed for when glucose is present. They're kind of waiting there and not using energy by translating. And then as soon as glucose comes back, they can start being translated again. And so we wanted to test that on a very long gene. Just looking at the chat. We wanted to test this on a very long gene. So FAST1 is like 6,000 base pairs long. And so that gives us time to know that if we, if we look for 5 minutes in plus glucose, we get very high luciferase readings. If we do 5 minutes in minus glucose, it's very low readings. 5 minutes is not enough time to translate for a new ribosome to bind onto this RNA and translate the whole thing. All right. So if we have a 6,000 base pair RNA, that's 2,000 codons. So that's about 400 seconds that it would take to translate this entire thing at 5 amino acids per second. All right, so within 5 minutes, any luciferase reading we're getting has to come from pre-existing ribosomes. And so we can see that when we add glucose back, we get this large increase in luciferase that wasn't happening in the glucose starvation. So we think this is further evidence that these ribosomes are stalling, and that the polarity is indicative of stalling for us, and that whenever we add glucose back, those ribosomes can kind of resume their translation. All right. So we have kind of these 3 different fates of RNAs, where these heat shock proteins, which again we said were very important for stress, combating stress, they have high initiation and high elongation and robust protein production. The hexokinase 1 gene and some of these other genes that we see very high transcription on, but very poor translation. I didn't get into their function, but as you guys asked about function before, these are alternative glucose metabolism genes, and similar to PGK1, we think that they may not be needed during the glucose starvation. But we have some other data that maybe they could be useful soon after you add glucose, and that by transcribing them and having initiation start but not having elongation, they might be primed for future glucose readdition. And then we also have these pre-existing mRNAs that seem to have low initiation, but also low elongation. So they maintain ribosomes on their open reading frame, but they have poor protein production. But that they can still, once you add glucose back, you can get robust protein production upon glucose readdition. So we wanted to try and explore kind of potential mechanisms controlling this elongation control. And so one protein that we're interested in is DHH1. And so it's an RNA helicase that's been implicated in slow ribosome elongation. Essentially, people have tethered it to RNAs, and even though they see much lower protein levels, they see that the RNAs reside in the higher polysomes. And so this sounds very similar to what we're seeing, where we get less protein production, even though we know that there's ribosomes present on those mRNAs. And so we wanted to look at kind of how DHH1 may be related to this. And so we took this polarity measurement of all genes and we looked at the cumulative distribution of those polarity measurements. And what we see, as I mentioned before, is that things are shifted to the right. So the delta polarity is generally positive for most genes upon glucose starvation. But previously, people have identified which genes DHH1, the protein, interacts with, or which mRNAs it interacts with. And so if we look at mRNAs that have low amounts of DHH1, uh, those genes actually have more negative polarity. Where the genes that have the highest amount of DHH1 are also the ones that have the highest polarity, which we think is indicative of the slowest elongation. And so another thing we see is that DHH1 overall shows like a length dependent recruitment during glucose starvation. So the longest genes have the most DHH1, but that may be just because those long genes are the ones that are accumulating the ribosomes over time. Like I mentioned initially, that the longest genes have the highest ribosome occupancy. And so we wanted to subdivide these into different mRNA length bins to see if there's a certain length where DHH1 becomes recruited. And so we looked at the ribosome occupancy versus the average. And so what we can see is that the top DHH1 genes have more ribosome occupancy for each length bin, while the bottom DHH1 have lower ribosome occupancy. So we think that this higher ribosome occupancy is because the ribosomes are stalled and they're not running off. But for the shortest genes, we don't see this correlation. And we think that there is enough time for the very short genes for the ribosome to run off. And so then we don't see any correlation between mRNAs that are highly bound by DHH1 and mRNAs that are poorly bound by DHH1 and their ribosome occupancy. And so kind of to add to what we're thinking is, we think DHH1 is enrichment is causal for this slow ribosome movement. And so RNAs that recruit more DHH1 end up having more slow ribosomes and a higher positive polarity. And so overall, we see that these genes that are the red genes, which are the ones that are robustly translated, on average have lower DHH1 binding, even corrected for their length, because there was some issues that they are in general shorter, and we know that DHH1 has a length dependent binding, but even corrected for the length of the gene, we see less DHH1 on these genes that are well translated, that have robust elongation during glucose starvation. The final thing we wanted to do is test what happens if we force DHH1 to an mRNA. And so we saw that genes like HSP30 have low amounts of DHH1 normally and are robustly translated. If we add a stem loop that we can drive recruitment through this RNA binding protein tethered to DHH1, we can force DHH1 to go to the RNA. And when we do that without the DHH1 present, we have robust protein production. If we force DHH1 to be localized, we see a decrease in RNA localization. All right. So that's kind of where we are with this story right now. And this is the lab where we use a lot of different techniques to look at kind of how gene expression is controlled and the impact that has, and trying to combine kind of, again, because we have all this sequencing data and trying to analyze it. A lot of people, when they're analyzing sequencing data, like ribosome profiling data, just get kind of the total counts for an mRNA. But we think this distribution of where the ribosomes is, this polarity score, is kind of indicative of the ribosome movement along the mRNA. So one of the projects that we've had some undergrads working on is this is in minus glucose, and we see this shift towards positive polarity. What about other stresses like heat stress? We've looked at osmotic stress. And what we found is that we don't see this same polarity shift in these other stresses. But there's a natural way for cells to glucose starve. If you just let yeast grow for a while, they'll use up all their carbon source and they go into some stationary phase. And what we've seen, this is also, they end up out of glucose. And we see the same shift towards positive polarity. And the interesting thing with that is that the dynamics we're working on here are like 15 minutes where we're seeing this buildup of ribosomes in the 3' end. But the stationary phase is over like 24 hours that they run out of glucose. So we were kind of very surprised to see this same dynamics distribution of ribosomes with the high positive polarity during stationary phase. And we're trying to understand, with potentially trying to model ribosome movement, how that could be possible over this longer time scale versus the short time scale where we have a little bit of runoff and then the ribosomes pause. Are they paused there for hours at a time? And that's something we're not really certain and we're interested in following up. I guess that's what I have on that. If you guys have some questions on that, I would be happy to take any questions. I could also briefly talk about kind of a different type of data that we're working with that is also using kind of computational analysis to get at. But first, I want to open it up if you guys have any questions. No? All right. So let me.
Oh, I have one real quick. Sorry.
Sure.
I wanted to think about it before I said it, which is always a good thing. Did your lab look at any possible byproducts of glucose metabolism being present and influencing the outcomes?
That's a good question. No, we haven't. So while we think DHH1 could be a contributing factor, I guess we don't think it's the only factor. And so we are still interested in potential other mechanisms. And I guess there are, potential metabolic changes, many metabolic changes that are taking place during glucose starvation that could be affecting this. Another thing we're wondering about is just this drop in ATP and GTP that takes place. How much of an effect does that have on translation elongation as well? So we've started coming up with quantitative reporters of translation elongation, and we can see a significant drop in elongation with these reporters during glucose starvation. But yeah, right now we haven't went further to find other factors that could also be contributing to this. And yeah, metabolites are super interesting and exciting, but also super difficult to follow up on.
Yep, cool. That's all I got. Thanks.
Cool. All right, I can do this one, I guess, more straight chalk talky. So another thing we're interested in, so I mentioned that we're interested in kind of post-transcriptional processes. And so one of the things we're interested in is the mitochondria. And so in red, we have mitochondria labeled with fluorescent microscopy. And these green dots you see going around that are actually mRNAs. And these are single mRNAs. And so this is a postdoc in the lab, Tatsuhisa. He's able to track these single mRNAs as they kind of are localized or not localized in the mitochondria. And then he can quantify kind of a localization ratio. And so TIM50 is a protein of the inner mitochondrial membrane. TOM22 is a protein of the outer mitochondrial membrane. But all of these, both of these are, the DNA for these genes is in the nucleus. And so these are nuclear encoded, but then their protein products are found in the mitochondria. And so TIM50, if we have like a localization ratio for TIM50, it has a very high localization ratio. While TOM22, you can see it's generally just like diffusing all around the cell. If you watch this RNA moving, it's diffusing all around the cell, and it doesn't seem to associate with the mitochondria. And so we can use image analysis software to quantify the location of every mRNA. And so we were able to do that. And so this is TOM22. And one of the things we're now trying to do is to come up with a model for this localization. So if we have TIM50 is highly localized, one of the things we found, and other people knew this, is that how it's localized, let's say we have a mitochondria here, how it's localized to the mitochondria is you have a translation, you have an RNA that's being translated, and there's a peptide that comes out which is bound by the mitochondria. And so there's a peptide signal called a mitochondrial targeting signal. And so this is what brings the RNA to the mitochondria. There's not a signal specifically in the RNA. Well, there are some RNAs that have like RNA binding proteins that will bind and bring the RNA directly to the mitochondria. For something like TIM50, it seems like it's completely encoded by this nascent peptide MTS. And so we found that TIM50 has another feature that helps it be localized. And so that's that at some point the ribosome stalls. And so then if we have an RNA and it's producing a peptide, we have some place where the ribosome gets passed and then that RNA is competent for binding. So once the ribosome passes point X, it's competent. And if it's not, I think someone's dropping off a package. And if it's hasn't passed that place, then it's incompetent. Hold on, someone's knocking at my door. I'm in a meeting, I can't talk right now, sorry. Oh, that was weird. They don't usually keep knocking. So one of the things we're trying to do is come up with a model for localization to the mitochondria, because we have another gene which is part of ATP synthase. It also has an MTS. So both of these have MTSs. But if we looked at the localization of TIM50 versus ATP3 versus TOM22, so TOM22 has no MTS. ATP3 has an MTS, but it has lower localization, and TIM50 is highly localized. And so right now we think that that's because there's a translational stall here after the ribosome has moved far enough. So now it stalls whenever the ribosome is kind of hanging out or the nascent peptide is hanging out of the ribosome, and it has more time to kind of find the mitochondria. And so we're trying to set up a kinetic model where we set the initiation rate, set the elongation rate, and have the RNA degradation rate. And can we then find a simple model where we have a mitochondria and we have different zones? And can we, by taking these parameters, show that we expect TIM50 to be more localized to this area than we ATP3? And I can say that right now, from the initial simplified model, it didn't work for us. And so we're having to incorporate some other factor to understand what is driving kind of some of this differential localization to the mitochondria. And so again, this is a project that we're taking in both image analysis of some of these microscopy data, but then we're also trying to set up kinetic models to help us understand some of the parameters that are driving mRNA localization to the mitochondria. And then we're also trying to do this not just for a couple genes, but for there's hundreds of genes that are localized. And so we're trying to mine through other people's proteomics and RNA-seq and ribosome profiling in order to come up with these initiation parameters, elongation parameters based on the protein levels, the ribosome occupancy, and the number of mRNAs. So yeah, I just wanted to kind of touch on that because it's kind of a very different kind of direction that we're pursuing, but it still all has to do with post-transcriptional regulation. And then we're looking in different conditions as well, and how the mitochondrial morphology changes, and how mRNA localization changes in those conditions, and whether we can also predict that based on these simplified parameters for RNA localization. All right, so any questions on this, or questions in general on kind of research or anything of that sort? I've had I don't know, a few undergrads, like, so we published a paper on this TIM50, TOM22, some of this RNA localization stuff a few months ago, and there was 3 undergrads on that paper, and most of them helped out more on the computational side. So I think we get a lot more biologists, biochemists in the lab, and then it seems like we've recruited undergrads to help us with some of the more computational analysis of some of our projects. So how many of you are doing research already or interested in research?
I'm doing research at Salk in the bioinformatics, the biophotonics core. We're doing like a project tracking mitochondria like dynamics and how they move.
Yeah, no, whenever I first saw this, I was surprised at how active they are. And again, like, some of these things look so cool. Like, why is my pointer? I don't know, like this looping that happens up here. Yeah, I don't know. It's really cool stuff. So yeah, so you're helping with the image analysis and kind of computational pipelines to analyze this stuff.
Yeah.
Cool. Anybody else already doing research?
It's not. Oh, go ahead.
Oh, sorry. I was just saying that I'm researching at La Jolla Institute for Immunology, like studying the 3D structure of the male X chromosome. But I'm looking into maybe a change of place.
So like doing Hi-C analysis type stuff, or? Yeah.
Right now my task is like turning, like visualizing the Hi-C data.
Yeah. Interesting. James, you were going to mention something?
Oh yeah, I'm transferring to either UCSD or SDSU this semester, but I got a NASA internship, and we're studying the microbes on the space station. So trying to figure out what abundances of different types of microbes are up there, and like kind of like suggesting built environment analogs. And hopefully doing some genomic analysis, like really basic baby stuff, because I am a beginner at Python. But,
yeah, I mean, the nice thing, I still don't consider myself a programmer, but I feel like I can make scripts do what I want them to do because there's just so much out there that I just Google, all right, how do I do this? And then put it in my script, and then it runs at least. And then I have undergrads come and tell me how horrible my code is, but it runs. So I'm usually happy enough. So do they do PCR up there?
Yeah. There's they do shock metagenomic sequencing and like 16S RNA sequencing.
Yeah, that's cool.
Yeah, it'd be interesting. I started like less than 2 weeks ago, so I'm just forming the project plan and everything, but it's definitely not, La Jolla Institute for Immunology stuff. It's wholly different direction, but I'm still excited.
Yeah. Oh, cool. All right.
Professor, so last week this Sunday we had a journal club and we were discussing one of the review papers from your lab. And we were actually discussing the paper, Post-Transcriptional Control of Mitochondrial Protein Composition in Changing Environmental Conditions.
Yep.
So one of the students actually had really wanted to know about this part. It's the mRNA localization part. I think you've discussed two mechanisms for the localization. um, so one was basically the zip code method, in which like the RNA binding proteins come there and um, bind to the RNA. And then the other one was nascent peptide tethering, I think. Am I correct in saying that? Like, so either a protein binds to the RNA and moves it, or like a peptide. Like when you say tether, as in doesn't that like you have the protein on mRNA, or does that tether to the ribosome?
So it tethers to the mitochondria, but if the nascent peptide is still associated with the ribosome, then that drags the RNA with it. And so we think that's why translation elongation speed is very important, because if it pulls the ribosome, but then things are elongating very quickly, then the ribosome's already elongated off the mRNA, and then the mRNA is not pulled with it. And so just because the peptide is going to make it into the mitochondria either way, because that's where the protein needs to go. And so it's just whether it pulls the RNA along with it. And so we found that translation elongation seems to be important. The other way we found that you can bypass this is if you increase the fraction of the cytosol that's mitochondrial. And so if there's more mitochondria, then that nascent peptide is going to find the mitochondria faster. And so then you can have fast elongation and still pull the RNA just because you find the mitochondria so quickly.
I see.
But there's a lot of unknown things. So one of the things we're trying to cook up right now is using like next-gen sequencing to do a genetic screen to identify mechanisms controlling both the RNA binding protein methodology and the nascent peptide localization, because it's been very hard to do like a microscopy based screen on 5,000 genes. And so there's new technologies using sequencing where you can use CRISPR and then put some barcodes in your CRISPR guide RNAs, and you can identify what guide RNAs are potentially affecting the mitochondrial. Well, this is in theory. We haven't done it and no one's ever done it. So we're trying to come up with a pipeline to do that using sequencing and then some analysis after that. But I guess were there further questions on the two different modes of localization?
I think his question was also about like how you differentiated using translational inhibitors between the two mechanisms exactly.
Yeah, and so the RNA binding protein, if we add a translation initiation inhibitor, it shouldn't be
Mhm.
affected by that. Where the
Of
nascent
course.
peptide, if we add a translation initiation inhibitor, so there's no new nascent peptide, should be
Right.
completely shut down by that. We
I see.
didn't go into as much, like in our
Review.
experimental paper, we didn't look at the RNA binding dependent localization as much.
Mhm.
But there's other papers that have tried to do this genome wide where they get rid of the nascent peptide and see, does that reduce localization? And so for 100 genes it does, another 100 genes it doesn't. And so that's why we think that there's these two different modes of localization. Why there's two different modes is a little unclear to me. Why didn't they all just use the same methodology? But yeah, I don't know. I guess that requires
Yeah,
a bit more studying.
that's very like gray area, right? So
Yeah.
much it's pretty new. It's interesting.
Yeah, and then also trying to understand things in terms of evolution is always super hard because yeah, it's just billions
Hmm.
of years of evolution, and then us trying to figure it out in a short period of time can be hard. But I think it's important to think about these things on why, and like you guys were bringing up, what is the function of these genes? And so I think those are all important questions to be thinking about when you're trying to connect your science to bigger picture things.
Yeah, like whenever I see a gene, like I want to know its function because it's usually the function that creates a difference in its levels, right?
Yes, I mean, yes, usually, I mean, there's people like Vamsi Mutha has used bioinformatics of co-transcriptionally regulated genes to say, oh, this is an unknown mitochondrial gene because it's regulated exactly the same as all these other mitochondrial genes. And so people have used just co-regulation to kind of specify function for proteins that we don't actually know their
Awesome.
function.
Well, that's another interesting topic.
Yeah. Yeah. So how long have you guys been doing this bioinformatics kind of chalk talks and journal clubs?
The journal clubs just started this Sunday, actually.
Yeah, we
Or,
started with
yeah.
your paper actually.
All right.
And then Chalk Talk's been happening at least the last 2 years from what I know. I don't know how long before that because I just joined UCSD last year. So
And if I have some undergrads that are semi-interested in bioinformatics, I should just give them your email and have them
yeah, for
reach
sure.
out
You
to
can
you.
give them our email and then we usually like communicate through a Discord that we can give them if they just shoot us an email.
Yeah. All right. Sounds good. I'll see. I know that, again, there's some that are starting that have some programming experience, and that there's others that are more experienced that are doing bioinformatics, and I don't know how busy all of them are, but I know it can be very busy in undergrad while trying to do research and everything. But yeah, I'll reach out to them and see if any of them are interested.
Yeah,
Well,
absolutely.
in that case, I would like to mention that at UBIC, we also have this another event called Bootcamp, Bioinformatics Bootcamp, where we kind of like go over many Python lessons and like basic machine learning lessons and phylogeny and alignment. So those are pretty cool if someone's
Cool.
interested
Yeah.
in.
How often do you do the
Uh,
boot camps?
well, we recently actually shifted to weekly.
Oh, all right.
Yeah, but there are lessons that are out there. So if like someone wants to like just yeah go through
Yeah,
them,
but
that's
it still
also
helped. I mean, especially with like with the pandemic, I had a student who was completely experimental, and then she was signed up for a summer program and she couldn't do any experimental work. And so then she had to go and find all these resources. But yeah, that'd be good to know if like there's local resources too that could help. And like, yeah, her learning TSCC, how to get on the TSCC account, and yeah.
yeah, definitely.
All right, yeah, I'll definitely forward this on to all the undergrads and see what students are interested. Because yeah, it seems like a good resource and a good way to learn.
Thank you, Professor.
Thank
That'll
you.
be very helpful actually.
Yep.
Cool.
Thank you. Okay.